All right. We're at 2 minutes in, and I do want to respect everybody's time, so we'll get started. So welcome everybody to join us today. My name is Zhiying. I'm the Director of Life Science and Technology at GemPharmatech US. I'll be the moderator for today's event, and I'll be introducing my colleague, Santi Chen, who is going to be the speaker for today's webinar. So to start, let me provide a brief intro of Santi. So Santi Chen, Santi is a research scientist and business development professional with over 10 years of experience in autoimmune diseases, immunology, and oncology. She received her training in human B-cell development at the Garvan Institute of Medical Research, Australia, in immunology, and followed by a postdoctoral fellowship at the Children's Cancer Institute, also in Australia, which is focusing on pediatric leukemia and therapies. Her projects included efficacy evaluation and mechanistic investigation of a variety of testing agents geared toward pediatric cancer. Her research projects resulted in multiple publications in journals such as Blood and Journal of Immunology, as well as national and international awards. Santi joined us last year, not very long, as Associate Director in Immunology and Oncology. We're really happy that Santi will join us, join our US team at GemPharmatech, and today I'm really happy that she's going to share her knowledge and experiences in immuno-oncology and oncology with us from a genetic engineered mouse models and the preclinical services perspective. So without further ado, I'm going to ask Santi to provide today's webinar. The title of today's webinar is Immunodeficient Mouse Models for Cancer and Immunotherapy Research, NCG, and the Next Generation NCG Mice. Thank you, everybody, and I'm going to give the stage to Santi.
Hi, everyone. Thank you so much for your time today. I'm very pleased to have this presentation today about our NCG and Next Generation NCG. So I will start sharing my screen, and I'll turn off my camera just to save the internet bandwidth. So today I will talk about the human immune system reconstituted NCG models for immunotherapy. I'm the Associate Director of Immuno-oncology at GemPharmatech. So humanized mouse model, what are they? They're invaluable preclinical research tool that enable researchers to perform more translationally relevant studies. Humanized mice contain functional human genes, cells, and or organs. Obviously, there's a lot of rapid advancements of cancer immunotherapy, and this year we celebrated 10 years of the CAR-T, the first CAR-T that was administered into human, and the person remains cancer-free. So there's a lot of interest in this area, and here we can see the various agents, and this is only a limited version. Obviously, there's a lot more companies working on cancer immunotherapy. So what is the need? There's obviously a lot of needs for more and more models that's more relevant and can reflect the human condition better. So there's two kinds of humanized immune model. So the first one is the immune reconstituted model, where the human PBMC or CD34 HSC is engrafted into severely immunodeficient mice. In GemPharmatech is the NCG mice. So there's a human target and the human immune system. Second type of humanized immune model is the genetically engineered mouse model, or we call it GEM. For example, it's humanized immune checkpoint models where the mouse still retains the complete immune system. We just engineered the human target. So there's knocking in of the human target. For the purpose of today's presentation, I will focus on number one, which is the immune reconstituted model. So a little bit of outline for today's presentation is we're going to talk about the NCG mice, severe immunodeficient mice, which is the backbone of GemPharmatech's immunodeficient model. And then I will talk about how this mice is being used with human PBMC reconstitution model as well as human HSC reconstituted model. I will also talk about the advantages and disadvantages of these two versions. And then I will talk about the next generation NCG, which is obviously with this model at the top here, there's some pros and cons. And we at GemPharmatech continuously want to improve our NCG lines, and we generated the next generation lines to improve the various deficiencies. So I'll talk about the NCG IL-15, which is to improve the NK cell, NCG SGM3, which is to improve the myeloid cell reconstitution, NCGX, which has additional mutation so that it doesn't require any irradiation, as well as the NCG MHC class 1-2 knockout to improve the human PBMC engraftment. So we'll get started with the NCG, which is the backbone of our severe immunodeficient mouse portfolio. In this diagram here, you can see from wild-type mice with a complete immune system, and over a period of time, various companies and institutions has generated the immunodeficient portfolio. And at GemPharmatech, again, as I mentioned, we use the NCG line, which is developed by GemPharmatech. And over time, we have developed the more than 20 next generation lines. So a little bit of background of the NCG. It is a NOD background, which has macrophage indecent deficient. We then genetically engineered the PRKDC knockout version that drives the absence of T and B cell, followed by the IL-2 RG knockout, which makes it deficient for IL-2, 4, 7, 9, 15, and 21, which makes it absence of NK cell. So the NCG is developed by GemPharmatech, and it is exclusively distributed by Charles River in North America and European markets. But for the next generation NCGs, it's available at GemPharmatech only. So the first use of the NCG line is to reconstitute it with human PBMC. PBMC is specialized immune blood cells with brown nuclei, for example, monocyte, lymphocyte, macrophage. It's developed in the bone marrow and located in the peripheral blood. Therefore, it's called peripheral blood mononuclear cells. So PB stands for peripheral blood. It's isolated, obviously, from the peripheral blood, the whole blood. To reconstitute this model, it's extracted from adult, and then we take the PBMC out, and then it's injected into NCG. Depending on the use, sometimes for evaluation of cancer agent, then the cancer cell may be inoculated shortly after the PBMC engraftment, or sometimes together with the PBMC. So this resulted in the human PBMC NCG bearing tumor for agent evaluation. So some features of this model, it's fast reconstitution. So within 1 to 2 weeks, it reconstitutes approximately 90% of T cells. A very short lifespan, about 4 to 7 weeks. So this is one consideration as you're designing a study on what is the best model, best cell line to choose. They do develop GVHD depending on the donor. It can be earlier, it can be later. One of the benefit of PBMC is the donor. Each donor can support about 200 to 300. Obviously, PBMC comes from adults, so there is a large volume that can be used to inoculate many mice. As I mentioned before, they do have short lifespan. Here you can see that on the right, survival rate here, it develops GVHD relatively fast, within 4 to 6 weeks. And as you can see here, the survival rate decreases over time. We do see this as well, looking at the pathology. Here on the left-hand side, we have the NCG, and on the right-hand side, human PBMC NCG with GVHD. And you can see that there's a lot of immune infiltration to the heart, liver, and spleen. Some pictures here at the top is normal mice, and down the bottom here, we can see the GVHD with the hunching and piloerection happening in the mice. So in terms of the reconstitution level, here at the top, we can see the CD45 positive cells. In the first couple of weeks, it usually reconstitute to about 90% of T cells. So even though the CD45 here remains low, but you can see the CD3, the T cell population, already reaches to 90+%. And this remains over the subsequent weeks. Looking further into the T cells, you can see there's consistent CD4, CD8, and some Treg population as well. So what can this be used for? This is an example of a case study where the PBMC NCG is being used with MDA MB231, so breast cancer line, as well as the RKO, so colon carcinoma line. And on the left-hand side here, it's testing with Tecentriq with a couple of doses. You can see this nice tumor growth inhibition. And the right-hand side, again, also with Tecentriq, you can see this tumor growth inhibition. Another example, this time is using a T-cell engager, a bispecific of CEA and CD3. So this one is, the study was conducted at GemPharmatech, and it's already a published study. The PBMC and the LST174T, which is colorectal carcinoma, is injected together. And then not shortly after, the agents begin to be injected and evaluation begins. The agent was tested across various concentrations, so 1 milligram and 3 milligram, and various version was tested. Here you can see it's as if there's only control and one agent, but actually all of the agents showed amazing tumor growth inhibition. And looking at it individually, here you can see that each of the agent showed very nice tumor regression. Another use of the human PBMC NCG model is for anti-GVHD therapy. As I mentioned earlier, this model do experience GVHD relatively early. So it's a great one to use to evaluate anti-GVHD therapy. And here we can see abatacept was tested at 2 different concentrations, 100 microgram and 500 microgram. And you can see that the survival curve improves with increasing abatacept. So moving on into another use of the NCG model is to reconstitute it with human HSC. So HSC is a hematopoietic stem cell, and it is a primitive cell that can develop into all types of blood cells. It's found in the peripheral blood, bone marrow, and umbilical cord blood. It's commonly used in transplantation therapy. The reconstitute, it will take approximately 8 to 10 weeks to reconstitute approximately 25% of CD45 in the peripheral blood. And the main immune cell that will reconstitute is T cells. One of the benefits of this model is the long lifespan of approximately 89 weeks. So how do we reconstitute this? We have the human CD34 HSC. The NCG mice will be irradiated. And then the HSC can be injected, followed by the cancer cell after the CD45 reaches to approximately 25% in the blood. Then we have the human HSC NCG bearing tumor, and then this can be used to evaluate cancer immunotherapy. Again, as I mentioned earlier, this model does have a long lifespan, so that gives you a lot of time to evaluate and monitor the agent that you're interested to test. So in terms of the reconstitution, here we can see the CD45 expression, CD45 in the peripheral blood. By about 10 weeks, it reaches already, for this particular donor, it reaches approximately 50%. The CD3 cells, it increases over time, which is a very standard process. And then you can see the CD4 and the CD8 remain consistent. As I mentioned earlier, this model is primarily T-cell driven. So you can see that the CD19, the B cell, decreases over time. And particularly the NK cell as well, we don't see a very low NK cell population. So an example of evaluating a T-cell engager using this model, in this case, it's using Raji, which is a B-cell lymphoma. And it was tested with Tri-TE and Blincyto, which is a bispecific antibody. And as you can see here, there's nice tumor growth inhibition. So I've described to you how to use the NCG model for cancer immunotherapy analysis, whether it's using human PBMC as well as human HSC. Obviously, the need is dependent on the study objective. If you use the human PBMC, the study duration tends to be shorter due to the GVHD. Meanwhile, for the human HSC, the study window will be longer, and it can reach up to 89 weeks. So I'll move on to the next section, which is the next generation NCG. This will improve some aspect of the NCG itself. So I'll start with the NCG IL-15. So why do we develop the NCG IL-15? As I mentioned in the previous slide, that in the NCG itself, once you inject the human CD34 HSC, there is really low NK cell. So it's not the ideal model for to study the NK cell. But NK cell is very important, and particularly when you evaluate an agent that requires ADCC. NK cell needs IL-15 to reconstitute, and that's why we knocked in this model with the NCG IL-15. So one of the first things that we do when we generate this model is to ensure that the IL-15 is being expressed. And here on the right-hand side, you can see the mRNA expression. In black is the NCG, and in red is the NCG IL-15. We then confirm this by the IL-15 protein expression. On the right here, you can see the NCG, as well as the NCG heterozygous in red and NCG homozygous in blue. So we then want to make sure that the IL-15 does support NK cell in 3 different scenarios. First, we inject the purified NK cell. Second, we also inject PBMC, human PBMC. And third is human CD34 HSC. So the question with each of this model is to understand whether we will receive successful NK cell reconstitution. So the first one here is comparing between PBMC reconstituted versus purified NK. So this 3 here is the PBMC reconstituted, 2 weeks, 3 weeks, and 4 weeks. On here is the NK purified, 2 weeks, 3 weeks, and 4 weeks. So you can see that the CD45 increases over time, which is a very normal behavior. And in comparison to the purified NK, even though this is much lower in percent, but if you look at the percent of the CD56, which is the NK cell marker, it's much higher compared to the NCG PBMC alone. Obviously, PBMC is a mixed population, so it's not surprising that there is only a small population of NK cell. Meanwhile, in the pure NK reconstitution, because it's pure NK, then we would expect a much higher reconstitution of the NK cell. For the T cell, the CD3, you can see that there is a high percentage of T cells in this one. And because this is pure NK, obviously there's a very limited T cell in this category. So for the NCG IL-15, they do have long lifespan. And as you can see here, when we tested over 140 days, the survival is 100%. And for the reconstituted level using the human HSC, you can see that the NCG IL-15 in red compared to NCG alone, it engraft the NK cell much better. And the CD45 expression is relatively similar. And then the B cell decreases over time as expected. The T cell increases over time, again as expected, and then you see CD4 and CD8. So this is a great model for T cell and NK cell study. With all of the reconstituted model, we want to make sure that they're not only reconstituted, but the cell that reconstitutes still functional. So in this, we evaluate the expression of the inhibition and activation marker for NK cell. In this case, you can see the KIR3DL is still expressed in the NK cell, and KG2D and KG2A is also still expressed. Just another way of confirming, we engrafted with a tumor. On the left-hand side here, we have the NCIH1975, which is a lung cancer cell, and on right-hand side, GIMT1, which is the breast cancer line. And again, you can see a very nice inhibition. We then take this one step further, both in vitro and in vivo. So for the in vitro here, what we did was we take the NK cell that was reconstituted in this mice. We take it out, put it in a plate, and then we incubate it with rituximab or together with Raji, or in this case on the right-hand side with K562, and we still see cytotoxicity. Again, validating that the NK cells not only reconstitute in this model, but they remain functional and can work through ADCC. And on the right-hand side here is the in vivo testing with rituximab and Blincyto. So the human HSC anti-GL15 are excellent model for efficacy evaluation of anti-tumor antibodies. So I'll switch gear to the next model, which is the NCGX. This model has additional KIT mutation, which makes it hematopoietic stem cell deficient. This provides the benefit that it doesn't require irradiation before injecting the CD34 HSC. So this makes the operational side is a lot easier. Obviously, irradiation or any kind of chemoablation is quite technical process, and if it's not handled properly, the mice can die due to the process. So if we don't have to do that, it just makes everybody's life a little bit easier. So when we compare in black the NCG mice and in red the NCGX mice without irradiation, you can see that the reconstitution level occur much faster in the NCGX. And you can see the T cell also reconstitute quite nicely, and the B cell decreases over time. Again, the NCGX is a T cell driven model. So when we analyze it at week 13, so at the top here is human HSC NCG with irradiation. And down the bottom is the NCGX without irradiation. Here you can see that there is a better engraftment. And particularly on the first column here, the erythroid population is much higher in the NCGX. This gives us an idea that there is other application to this model, not only to evaluate cancer immunotherapy, but potentially the blood diseases such as beta thalassemia is a possible use of this model. So another model in the next generation NCG portfolio is the NCG SGM3. The S stands for SCF, G for GM-CSF, and 3 for IL-3. The combination of these three supports the myeloid cell development with human CD34 HSC reconstitution. So in these figures here, in black is the NCG SGM3, and in gray is the NCG, both with irradiation and both with human CD34 HSC reconstitution. And here we can see that the CD3 reconstitute quite nicely. You can see CD4 and CD8 expression cells is higher in the NCG SGM3. B cell decreases over time. NK cells decreases over time, and also plasma B cell. Most importantly is the myeloid component. Here you can see in the total myeloid cell, the black, which is the NCG SGM3, is much higher across 17 weeks. When we look deeper into the monocyte, granulocyte, neutrophils, DCs, M1 and 2, you can see across the board that the black bar, which is the NCG SGM3, is much higher compared to the gray bar. again validating that this model is an excellent model for myeloid cell reconstituted study. So an example on how to use this for anti-tumor agent evaluation. Here we have the A549 model in NCG SGM3 with human CD34 HSC. In this case, it was tested with anti-human PD-1 and anti-human PD-1 with agent X. So this is a fine agent, so we will not disclose what the particular agent is. So you can see here the tumor inhibition in the single agent or in combination doesn't seem to be too great. But most importantly is what is happening within the tumor itself. So we take this one step further and analyze the tumor infiltrating lymphocyte or TIL analysis. Again, in black is the vehicle control, in red is anti-human PD-1, and in blue is the combination. You can see quite immediately here, the Treg population is upregulated in the combination. There's reduction of plasma. Most importantly, again, looking at the myeloid component, you can see that there is upregulation of M1 and downregulation of M2 in the combination therapy. So this highlights that even though the tumor growth inhibition may not be too striking, but certainly this agent X is doing whatever that it needs to be, and it's highlighting the importance of studying the tumor infiltrating lymphocyte to see what is it doing within the tumor environment, tumor microenvironment itself. So finally, I also want to quickly touch of the NCG MHC class 1 and 2 knockout, which is one of our recent addition to the next generation portfolio. So this model have, obviously, as the name suggests, knockout of MHC class 1 and 2. The idea with this model is to improve human PBMC engraftment to reduce the GVHD occurrence, providing a longer study period. So I'll walk through this slowly. So in black is NCG alone, in red is NCG MHC class 1, 2, and in blue is the MHC class 1 alone, and in yellow is NCG beta to M knockout, which is another model that can be used for human PBMC. And here you can see with the GVHD scoring, there is a significant decrease of GVHD, particularly in the MHC class 1 and 2 knockout. And looking at the survival curve, again, this model can last longer in comparison to just a straight NCG alone, which can develop GVHD within a span of as soon as 4 weeks after PBMC graft. So just to give you a quick summary. So today we've discussed about the severe immunodeficient mice, the NCG library at GemPharmatech. I've also described how it can be used with human PBMC, but keep in mind that GVHD do occur fast in the human PBMC NCG model. Alternatively, you can also use the human HSC NCG, which will have longer study period. Obviously, there's pros and cons using each of the model. Therefore, we developed the next generation NCG, the NCG IL-15, which have the IL-15 knocked in, and it improves the NK cell reconstitution. The NCG SGM3 with SCF, GM-CSF, and IL-3 to improve myeloid cell reconstitution. We also have the MHC class 1 and 2 knockout to improve the human PBMC engraftment, and the NCGX, which will allow human CD34 HSC reconstitution without the need of irradiation. Last but not least, I just want to give this summary. So this is our recommendation on which NCG is best for which immune cell, depending on what is your immune cell research. So for example, if your interest is only the T cell reconstitution, so this is the models that we suggest. NCG, HELIS, beta-2M, IL-2, and also the MHC class 1 and 2. If you need NK cell, then let's move to the left-hand side here, the NCG IL-15. NCGX or NCGX-IL-15 is also another one that will be useful. If you need the myeloid component, then we suggest the NCG-SGN3. And as you can see from this graph, there are other NCG, next generation NCG that I have not discussed today. So on the B cell side, we have the IL-6 and the BAF and 6CL13. We also have other NCG that is being knocked in with, for example, here, the CD47, HER2, CD228, and some other next generation NCG that is useful for other purposes. So if you're interested in any of these NCG, I highly recommend you to get in touch with us. So our details is here. Or myself or my colleague Zhiying can help you to answer some questions. Thank you so much. I really appreciate your time, and I look forward to having the discussion.
Great. Thank you so much, Santi. I think now we can open the camera and let's see whether we're challenging our bandwidth by doing so. All right, so let me check into the Q&A. I'll read the question to you, Santi, so that you can have a break, a little breath. So the question is, do any of your models have functional complement to test CDC?
Sorry, can you hear me? I'm having a little bit of problem with my computer here.
Yeah, we can hear you now.
Do you mean functional? I'm not too sure I understand the question correctly.
Yeah, let me read it again, and hopefully, you know, Lena, if I'm not pronouncing your name correctly, please let me know. But I'm gonna read again, and Lena, if you can type to clarify what you meant by complement, let us know, or let Santi know. So the question is, do any of your models have functional complement to test CDC?
Do you mean the complement-dependent cytotoxicity assay? That's a very interesting question.
She said yes.
I don't. That's a very good question. I unfortunately don't have the answer to that. I don't see a reason why we cannot develop that. That's actually something for our internal team. Yeah, so I apologize. Zhiying, do you have an answer for that potentially?
No, to be honest, I have to say, like, I don't even know what CDC stands for at this point. So, Lena, if you want to continue the discussion, feel free to type in, and otherwise, we'll definitely pass it on to the R&D team. If we have an answer for you, we'll follow up to let you know. If we don't, we'll, you know, try to develop something in that direction. Right, I'm going to move on to the next question, which is, how do you HLA match your tumor cells with a human PBMC donor you use for engraftment in a human PBMC NCG mouse model?
I think for HLA matching, we can select the PBMC donor. So we can do HLA typing of the PBMC donor before using it in a study. Sometimes it may not matter, depending on the study objective. The HLA matching may or may not impact the study itself. But if it is an absolute requirement, then we can do HLA matching.
Right. So now I got a couple of technical questions. I'll read it out to you, Santi. So the first one is, why is irradiation required when you use human HSC?
So if you use human HSC, the radiation is required to suppress the immune system a little bit more. Meanwhile, if it's in PBMC, the PBMC itself is I guess already more mature, so they don't need as much suppressant. But because HSC is such an immature cell population, and for them to reconstitute well, that irradiation is required to suppress the host immune system to allow, and also to make space in the bone marrow to allow the for them to take over the compartment.
Right. Next one, also about the humanization or reconstitution. So when you do immune reconstitution, how many mice can a donor be used for?
For PBMC or HSC?
Didn't specify. Maybe you can talk about both.
Yeah, so PBMC, typically you can reconstitute about 200 to 300, so quite a large number of mice. But then again, when you're designing a study, you have to remember for every study, there's always average mice. So I don't recommend a study exactly the same amount as the maximum, because you have to take in count into the average. In terms of HSC, HSC is a little bit more limiting. You can get, depending on the various vendors. I know for GemPharmatech, we can do about 50 to 80 per mice. Again, depending on the donor and the mouse model that's being used.
Okay. I think this is probably a little more technical in a different direction. What kind of animal housing facility is needed for housing the NCG mice? Also, is there any special food, water requirements?
Well, I think that one is more directed to you, Zhiying.
Right, I'll take it. So for NCG mice, since they are immune compromised or immune deficient, either way to call them, they do prefer or live better if the environment is cleaner, the quote unquote cleaner. So it takes upon the health monitoring program as well as health testing to make sure in their environment there is no pathogens that can elicit disease in these models. So the in terms of special, I would say it's not special as you have to build something really new and no one has it, but it is depending on how you care for these animals. Food and water, typically people use irradiated food and autoclaved water for these animals, and in some facility such conditions are called the so-called sterile environment, but is a name, is basically means we are providing the animals with a cleaner environment compared to the typical SPF health standard. All right. I hope this answers the question and provides some perspective on how we can raise and care for these animals. I think this is the last question I have for the moment. So what should one consider when choosing PBMC versus HSC humanization when using NCG mice?
There's a few things you need to consider. Number one is the study objectives. PBMC does give you a much shorter study period. GVHD sets in within 4 weeks. Sometimes you may see it starting from 3 weeks. So the depending on the study, again, just keep that in mind. Meanwhile, in HSC, the study period is much longer. The number of mice that can be used is another factor. If you use PBMC, then you can engraft a lot more mice. HSC is a lot less mice. The number of donors to be used within the study is another important factor. Our recommendation is to not just use one donor because there is donor-to-donor variation. At the bare minimum, use 2 to 3. If you can do more, that would be great. But obviously, it's not easy to source the donor. In terms of the technical, more the scientific aspect, PBMC does contain, it comes from adults, so it does contain more mature cells. HSC is more immature, so hematopoietic stem cells. So if your study requires something that's a lot more mature, certain kind of cell type, then that's another consideration to take.
Okay, great. So I'm going to take a last look into the Q&A and make sure we get every question. All right, well, I think there's one that you already touched upon and sort of provided an answer during the talk, but let me just ask it again on behalf of the audience. Do these NSG mice, or actually this is NCG mice, develop host versus graft disease? If yes, for how long after the engraftment can we use them? You're muted.
Oh, thank you. If it is PBMC, then the graft versus host would happen fast. But if you're asking host versus graft, the other way around, if it is NCG mice, they are already severely immunodeficient, so that doesn't tend to happen. It's more the graft to the host, not the host to the graft kind of direction. So yeah, I hope that answered the question.
Okay, so if, let's say, graft versus host, how long after engraftment can they be used? I guess that's the real question.
For PBMC, study duration is typically within 4 weeks. You can get to 6 to 7 if you're lucky, but it is kind of pushing it. For HSC, reconstituted study, it can go up to 30, 40, 50 weeks. So it lasts a lot longer.
All right. And I don't see any other questions popping up. So thank you, Santi. And thank you, everybody, for joining us today. And we'll conclude for today's webinar, and looking forward to see you again in May. All right. Bye.
Bye, everybody.